Assalamualaikum and a very good afternoon out there to all the participants for today. And welcome to our INBIOSIS webinar lecture series 4. My name is Nurkhalida Kamal and I will be your moderator for today's webinar series. Before we start with our talk by Dr. Ahmed, let me introduce a bit about Institute of System Biology or INBIOSIS. So basically INBIOSIS was established 15 years ago as a system biology research center which comprising multidisciplinary team members which we always working on interesting biological questions. So for today's seminar is actually our 4th webinar for the series. And of course, it will be the final lecture for this year 2020. And we hope actually this seminar can be used as a platform to bridge and also share knowledge and experience in this pandemic time, which keep us active and also motivated in doing science research. So basically, the webinar will take about 1 hour, where Dr. Ahmed will give a lecture about 45 minutes, and after that we will have 15 minutes Q&A sessions. So participants and audience, you can write any questions during the lecture in the chat box, and then of course we will respond this Q&A, this question during the Q&A sessions. And before this seminar ended, we would like to invite all participants to on your camera for the photography sessions. And please stay tuned until the end of the lecture, where we will send you the link for the attendance form for e-certificates. And also for UKM staff, the ISPEL UKM credit. All right, now comes a very exciting time. It's my honor to introduce our speaker today, Dr. Ahmed Mediani, and let me share with you his brief biography. So basically, Dr. Ahmed Mediani received his bachelor degrees in agriculture and nutrition from University in Algeria. And then he obtained his master's and also PhD degrees in food science from the University Putra Malaysia or UPM. He is currently serving as a university lecturer or research fellow in the Institute of Systems Biology INBIOSIS in UKM. He has published a number of research papers in referred international journal, and his research area is in food sciences, metabolomics, and also food chemistry. So without further ado, I would like to invite Dr. Ahmed to share his thought and also about the science for this webinar series for today. Please welcome Dr. Ahmed Mediani.
Thank you very much, Dr. Khalida. Assalamu alaikum. Bismillah, salatu wassalamu ala rasulillah. Good afternoon to all. Thank you for the kind introduction. Today I want to present to you about the topic, the current, because it is based on the current situation, the metabolomics to study the COVID-19. As the Dr. Khalida mentioned, this is the final for this year. Inshallah, I will be okay. The topic is based on the current situation about the COVID-19. The topic is when I choose this topic, I thought there is so many study on COVID-19 using the metabolomics, but then when I search, there is few studies. Later at the end of this presentation, I will share with you few examples. Let me start by introduction. The background on the coronavirus. The coronavirus is named SARS-CoV-2 or COVID-19, is a virus that is common in people. The most common reported symptom including fever, dry cough, and tiredness. The people, when they have the COVID-19, mostly they will have fever in the beginning. That's why when you go to the mall or something, they will check the fever. Or sometimes they will have dry cough or they are tired. And in the mild cases, people may get just a running noise or sore throat. Some people they will not notice they have COVID-19. And in the most critical cases, people with the virus can develop difficulty breathing and may experience organ failure, and some of the cases are deadly. Some of the studies that have been done on the COVID-19, they have focused on its epidemiological and clinical characteristics. And about 80% of the patients infected with COVID-19, they have mild symptoms. They usually recover without any medication. Therefore, they are classified as mild or moderate COVID-19. However, 20% of the patients, they suffer from respiratory distress and they need immediate oxygen therapy. And they need to be in the ICU, in the hospital. They need to be hospitalized. And for the studies, many immunologists and virologists are currently searching for the COVID-19 from the point of view of the gene expression. That's why many studies, they focus on the gene expression because they think they are based on the analogy that your DNA is the cookbook. They just follow they think the gene, and sometimes is used by the people, and the product of the recipe and eventually product of the gene is of the body is made of protein. When we compare between the omics and metabolomics and with other omics techniques and the metabolomics, and basically when the people think about the COVID, sometimes they just think about the genomics or the transcriptomics or proteomics. And this is the fact. When the proteome should be preferably be studied, but this is very costly and difficult to do. And the transcriptomic is better, and this analysis is typically done because it is the best way to get a detailed snapshot of the gene activity of the human, which is just a single step away from the reality of the protein, because most of the gene is made of protein. That's why they think that when they study the protein, they will cover this disease. However, the metabolomic can be very preferable because it provides a view that is a step closer to the fact than transcriptomics, because the metabolite, they are the end product of the gene expression. Later on, I will make it in detail when you see the comparison between the omics. For the omics technology, the holistic approach in the drug discovery in medicine, which is introduced, omics technology become the new approach which claims to be advanced technique rather than the classical one. The strength of omics technology lies in their abilities to profile various aspects of the drug discovery process in a large scale. They study all the metabolite and all the effect in one time. All the factors can be studied, and then they are compared. We come to the metabolomics. What is the definition of metabolomics? The metabolomics is a comprehensive tool for evaluating the metabolite variation of organisms under various conditions. The metabolomic can offer quantitative and qualitative that has been used for monitoring metabolic variation for in vitro or in vivo, for the compare the plant or the animal or human metabolic. Then the metabolome is the downstream product of the gene expression, like I just say just now, and reflect the functional level of the cell. Because sometimes when we see the study about the genomics and the proteomics, sometimes they will say there is no significant or there is significant. But when we go back to study the metabolite, we can see they say not significant, but when we see at the metabolite, we can see them as significantly changed. Or sometimes the protein can be significantly changed, but because of other factors that can affect the metabolite, they will not be significantly changed. Then when we compare between the the omics approach, the genomics can show us what can happen. And the transcriptomics, it will show what appears to be happening at that moment. And the proteomics, it will show what make it happen. But the metabolomics, it will cover all this and it will show you what has happened and what is happening before and after. That's why to study the metabolome is better. I'm not saying that metabolomics is better than other omics, but if we study them complementary to each other is better. Then we can have the whole picture of the changes. The benefit of analyzing the metabolome, the metabolomics has advantage because the number of metabolites lower than the number of gene and protein in the body. This one can make the sample reduce the complexity. And also, as I said just now, although the concentration of the enzyme or the metabolic flux may not significantly change during a biochemical reaction, but the concentration of the metabolite can be changed significantly. Even the minor one can show significantly change. And also, this one, the metabolomic reflect more accurately the functional level of the cell. And also, metabolic flux is regulated not only by gene expression, but also by environment, because other factors can affect the final product, the metabolite. That's why measuring the downstream product, the final product, as the metabolite is better. And also finally for the metabolic experiment are 2 to 3 times less expanded than the proteomics, and it is very fast. However, there is some challenges and problem when you do the metabolomics. The metabolite have wide range of molecular weight and large variation in concentration. As I say just now, when you study because sometimes some minor metabolite can do the changes. And also some metabolite, if you isolate them or you tested them, when you study the drug, if you study only one drug, it will not be effective. But the synergistic effect, sometimes when you do for all the metabolite, then it will show effect. And also, sometimes when you study, you see some metabolite, they are very minor and they are the VIP or they are very significant. And also, loss of various metabolite during extraction. The extraction is very critical step when you are dealing with the metabolomics study, and there is no method of extraction that can extract all the metabolite. So that's why sometimes you will lose some features. And the metabolome is much more dynamic than the proteome, which make the metabolome more time sensitive. For the metabolomics, the study is very critical. Also, when you harvest your sample in the morning or afternoon, the metabolite can be changes. That's why you need to fix, and also you need to check and compare between different time and see which time is better. And also the sample collected for metabolomic analysis require careful handling and information regarding physical activities and other patient validation. Sometimes when you are dealing with the diet with the human, when we give different human different diet, also it will affect the final product, the metabolite. That's why we need to be very careful. The metabolomics application. The metabolomic can be applied for bacterial characterization and also for human health assessment, for metabolic engineering, and also for identification of metabolic biomarkers that can change as indicator of the presence of the disease. That's why we can apply these metabolomics on the COVID-19. Application on viral infections. Also, the metabolomic can identify key biomarkers of the infection. Because the COVID-19 is viral infection, we can apply it. It can also identify the key biomarkers of the disease progression. That's why the mild and moderate and also the critical cases, we can check and compare them. It can also evaluate the effect of the vaccine treatment. Like now, we already have the vaccine for the COVID-19. We can, by metabolomics, we can check the effect of this vaccine. And also it can identify the biomarker of the therapeutic response. The metabolomics also can provide useful insight to research effort to this global pandemic, as we mentioned, for the application on viral infections. Through examining the metabolome, we may calculate the dynamic of the cell response to internal or external disorder in order to gain a deeper understanding of underlying biology from the cell. This information can promote the identification of biomarker and biological pathways that are active or inactive during the disease. We can suggest also the mechanism of action. Even when we give the treatment, we can check between different treatment. Now we have different vaccine. Also, we can check the mechanism of action of different vaccine, and also check the effectiveness of any of them. And this answer basically answer the biological questions and enable metabolic phenomena in the cell to be studied. Let's move to the metabolomics for COVID-19. The study of metabolic variation between the COVID-infected patient with critical symptoms and those with moderate symptoms can be tested. And also this is important to determine why any of the metabolic markers are correlated with the disease period and mortality. Like when we have the quarantine for 14 days, we can see what is the difference between from 0 to 14 days. Then we can compare and see the symptoms. And also differentiating between serious and moderate condition. This one can allow high-risk patients to get better target treatment, and also to give the treatment. And also this one can enable us to give a medical professional. This is where the metabolomic can offer a compromise, and this lies somewhere between the snapshot of the transcriptomics and the gene expression and the metabolomics. This one when we compare between the omics. For some studies that has been done before, this is they suggest these biomarkers as the glycoprotein, and this glycoprotein can be tested by the NMR, and this one made it as the correct option for the metabolomics. This glycoprotein already proven to be for the biomarker for the inflammation, and in the current study, they also correlated with the COVID-19. This is very interesting biomarkers used to study the COVID-19. And also the researchers now looking at lipoprotein particles and also fatty acid, amino acid, and several other options. Later on in the example of the study that I will share with you, you will see they already suggest some biomarkers. this is one of the examples. They did proteomics and the metabolic characterization of COVID-19. They by complementary the proteomics and the metabolomics. These two approaches were applied to assess which cases will likely become clinically critical. And these two technologies were performed to analyze the proteome and the metabolome of the serum from COVID-19 patient and several control groups. They compare between, here you can see, they compare between the healthy and non-COVID-19 and the critical and the non-critical patient. Then they did both proteomics and the metabolomics, and then they can suggest and see 894 proteins, and for the metabolite they can see 847. Here from the heat map, this is from the result of that study, we can see that they already compare between this type of the healthy and non-COVID and the critical and non-critical COVID-19 patient. And they see the metabolite that's contributing to each type of them, and also compare between the age. Sometimes they compare between different several ages, between 20 to 70, and see how the effect for each patient. And also they compare between the gender. For male and female, they find some biomarker different from others. And also for this is the metabolite, the one they already find in the result, and then they suggest which one is contributing to each model. Here they compare between the non-severe and the healthy, and they find this is the critical and the significant metabolite, and then they suggest them as biomarkers. And here they compare between the critical and the healthy one. Also they find the metabolite they are contributing to the critical. And for when they compare the non-COVID patient with the healthy, they find other compound they are contributing. This is what metabolomics can offer. And they suggest the mechanism based on the proteomics and the metabolomics as a complementary study, and very nice. And later on I will show you the mechanism that they propose. They detected increase in some metabolite, the essential intermediate for synthesizing corticosterone, suggesting that biosynthesis may be protective mechanism against COVID-19 infection. They also find evidence of significant activation of phenylalanine pathway, and also they enriched patient and nicotinamide adenine, the cofactor in minicellular redox reaction. The macrophage process is closely related to lipid metabolism. Over 100 lipids were down-regulated, which is they suggested as a biomarker because they are down-regulated. The data showed decrease in sphingolipid in both critical and the non-critical and the critical COVID patient, and they suggest sphingolipid and glycophospholipid are important compound of biomembrane which mediate signal transduction and immune activation process. They relate between the immunity system and also the inflammation to the COVID-19. They have almost the mechanism like the immunity system and also the inflammation. This is another study. This one they study this. They compare between the COVID-19 from other respiratory condition. This one is good because some people they will have just normal flu, and they consider them as a COVID-19. That's why they did this study, and then they did with the GCIMS and compare by multivariate data analysis. The potential COVID-19 breed biomarkers were identified by multivariate data analysis and compared to GCIMS database. The analysis identify ethanol, octanol, and ketones and methanol that discriminate COVID-19 from other conditions. They find this chemical as the biomarker for COVID-19 as compared to other respiratory diseases. Here we can see that the COVID-19, they are clearly separated from the other non-COVID, and they find the metabolite they are contributing to this separation. Here we can see that when we talk about metabolomics, the metabolomics is involve many fields, the biochemistry, the chemistry, and also the medicine, pharmacy. That's why we need the collaboration to do the metabolomics study. This one, when we are dealing with the metabolomics in the system biology, we need to deal with many fields and also need to have collaboration with other people to have critical study and also overall study to cover all the topics. That's why everyone can tell different story, and collaboration in metabolomics is very important. And in the final, don't forget that metabolomics is a tool, not a goal. And the metabolomics is a method to observe the biological system and to detect biomarkers that can be done for COVID-19. And also this one, the metabolomics can be used for COVID-19 as other diseases because it shows that very effective method to study the disease and to find the chemical marker that's related to many diseases. This one, it also can be used to study the COVID-19, and you cannot do it alone, as we say just now. That's why we need collaboration, and we have many experts in INBIOSIS, and you are welcome for collaboration with them. Thank you very much, and that's all for me. If you have any questions, I am very happy to answer them.
Thank you very much to Dr. Ahmed for very interesting talk that you share with us regarding metabolomics in studying COVID-19 patients. So to all the participants and audience, you are welcome to send your question in the chat box. While we're waiting for that happen, so Dr. Ahmed, I have one question to ask. Because you mentioned about the biomarkers, different biomarkers when you study metabolomics. So is there any difference in terms of metabolites biomarkers between Caucasian population versus Asian population? Because we know that COVID-19 infects many population in the world. So do you have any thoughts about that?
Yes, I think there is differences. Also, the environment, as I say just now, there is so many factors can affect the metabolite. Also, the environment from different country to another country, and also the diet. There is other physical activities also, and also between different people also can be different. That's why the biomarkers for different, also from different people, they can be different between Asian, European. Also, when you target the the biomarkers, also when we have the treatment, we need to think about different people and also see how the diet and also the physical and other factors rather than this. Do I answer your question?
Yes, of course. So meaning based on from your answer, what I understand, yes, of course, there will be a different biomarker in terms of different population in the world between, for example, Caucasian and Asian, because many factors that contribute to this infection towards the patients. So may I know, if you know, is there any active or ongoing metabolomics research study that, you know, focusing on COVID-19 in Malaysian patients?
I didn't hear there is a study here because I think that the human ethic in Malaysia is very difficult, and also there is no facilities yet here. I think only IMR they have facility, and also they are still not that advanced in this research. But for metabolomics, until now I didn't hear that in Malaysia there is any study has been done.
Right. So meaning is very important, I mean, for us as a Malaysian, you know, to start doing the study, metabolomic study on the COVID-19. That's specific.
Yeah, that's why today I
Malaysian
want to suggest for you to do this metabolomics, because the people now they are focusing on the genomics, proteomics, and other studies, because that one is very costly, and the metabolomics it's easier to done, and it can be done since they are dealing with the genomics and other omics. It's better to do the metabolomics if they have the facility. And no Malaysia already have the facility for metabolomics. And many other diseases related like inflammation and immune system already done. Why not the COVID-19?
Thank you, Dr. Ahmed. So there is several questions in the chat box. Let me read for you. So question from Ili Nadira would like to ask, how early or sensitive is the biomarker detection using metabolomics? For example, days of infection.
How early sensitive is the biomarker detection? That's when we can compare between different patients in different days. Maybe one patient we can see in different days and see how the metabolite differences, and the metabolite that has significantly changed, that's why we can consider that one is the biomarker. And from different, we can draw the conclusion after we test many patients, not only one patient. Then between different days and see how the differences. We can trust that biomarker after.
But from the current, I mean, findings on the metabolomics on the COVID-19, is there any proposed, I mean, days of, you know, early biomarker detection that any research group have been proposed, or there's still no?
The one I just show, they have many. The study, the one they did, they show the glycoprotein, the one can be done by the NMR, and also they have the amino acid and the fatty acid also, lipoprotein also.
So can we move on? I think we move on to the next questions. All right, so question from Nisha. So Nisha asked on single breath sample collections for COVID-based metabolomic analysis. So her question is, could you elaborate on the advantages and also disadvantages of this strategy for real-world application? So meaning, is it feasible or not feasible on doing this type of biomarker detection using the breath of COVID-19 patients?
Because the GC-IMS is dealing with the volatile compound, this one is very useful method to study and to compare between the different respiratory diseases. And COVID-19 is also one of the respiratory, it will affect the respiratory system. That's why it is useful to study based on this analysis.
Meaning, I think the question is maybe she wants to ask, is it feasible in terms of the setting, to do this type of biomarker detection or study, doing on using the breath of the patients, because it's very easy for us to be infected with this virus.
Yeah, because the virus can be transferred by the air. It's also useful to study by using the breath. I think it's a suitable method to study this.
Okay. Thank you, Dr. Ahmad. Okay, we move to the next questions from Sharul Natakain. So her question is, how you can contribute or elaborate your current research for COVID-19 study?
What was the question?
How you can contribute or elaborate your current research to COVID-19 study?
You can contribute because I study different diseases. I show different biomarkers of most of my study on diabetic, and I find then we can compare between the healthy and the one induced for diabetic. Also, this one can be done for the COVID-19. Also, we can compare between the healthy and the COVID and see what are the biomarkers between these. It's almost the same objective. This one can be applied
but
for
maybe specific on, Malaysian population, maybe.
But for the COVID-19 is mostly, it should be done on human, not on for us we are doing on the animal model, but for the COVID is supposed to be on human. And in Malaysia, it's very difficult to get on the ethics for the human. I'm suggesting this study, and hopefully it will be done here.
Quite difficult, but must be done, is it?
Supposed to be done.
Yes, all right. So we move to the next question from Kash Vinta. She asked two questions. The first one is, sample collection must be done how many times in order to get an accurate data? I think maybe how many replications that we must have for the metabolomic study.
Most of the metabolomic studies, they suggest more than 5 replicate, biological replicate. If 6 to 10 is better.
More than 5, 6 to 10 is better for one biological sample, for the replication. Thank you, Dr.
We
Ahmad.
can also have the biological replicate for more validation for the study.
All right. So for the next questions, and can we use other elucidation methods or identification methods rather than NMR? Is there any other methods that can be used for metabolomic study?
Yeah, for metabolomics can do based on the MS, LCMS, GCMS, rather than the NMR. Not only the NMR can be done, because most of the amino acid, fatty acid can be detected by the MS also. This is all primary metabolite that can be detected by the MS also.
All right. Thank you, Dr. Ahmed. So we move to the next question by Dr. Shahliza. Her question is, sample collection may result variations in terms of the analysis. So may I know how do you how do you do the sample collection for your study?
That's why we say just now, when you collect your sample, you need to collect it at the same time. If you do in the morning, you need to treat all the sample the same thing and collect it at the the same time. Sometimes when you have different groups, you cannot collect the sample for some group in the morning and another group in the afternoon. And also, if you are comparing between different days, not in the same day you collect the sample at 8 o'clock, but in the next day you collect it in the afternoon. This one you will have a variation. It will affect the metabolite.
All right. So meaning we need to be very specific, or need to mention the times, the technique that we use for sample collection, because this will affect the metabolites. All right, we move on
animal
to the next question. Okay, from Rohana from INBIOSIS, her question is, because we know that this COVID-19 virus can be infected in animals. Her question is, can plants also get infection of COVID-19 virus?
Can plants also get? I think this one.
No.
Because it's common in people, not in the plants.
Okay.
Animal or human?
An animal.
The plants, I don't think so.
All right. Okay, thank you, Dr. Ahmed. We move on to the next question from Muhammad Zuwairi. So in terms of the economics perspective, do you think metabolomics approach can replace PCR and rapid kit tests? And how fast actually the result can be obtained?
For metabolomics, especially when you are dealing with the NMR, it's very fast. The sample preparation and the analysis can be done in less than 30 minutes. And also the sample is much cheaper than the kit of the PCR. That's why when we compare the metabolomics and the other omics study, we say it's 2 to 3 times less expensive than the other omics. And also,
perhaps, metabolomics technique can be used as a complementary technique,
yes,
other than PCR or kit test. Rather, we replace it.
Build the model. We need first we need to build the model. Then, when we have the sample, we can just compare, and we put it in the model. Then we can detect. It will be easier for us to compare.
All right, so we have another last question, I think, for this from Rafida. So her question is, in case of COVID-19 for metabolomic study, so do you suggest to use targeted or untargeted approach, and why?
This is depending on your objectives. If you already know the biomarkers, and also maybe if you want to correlate it with the inflammation or immune system, and you already have some idea about the biomarkers and you want to check them, maybe you can go for targeted. But if you want to just profile all the metabolite and see which one has you can go for untargeted. You can use the NMR to see all the compound, and you can suggest your own metabolite and biomarkers.
All right. Okay, thank you, Dr. Ahmed. And okay, we have another 2 questions because we still have time, like 10 minutes. I think it's okay for us to ask you more questions. So from Ibrahim Jantan, his question is, metabolomics use spectroscopy methods such as NMR and mass spec supported by databases which are predictive methods. What is the level of accuracy when these data are used to interpret the underlying mechanism of action?
The mechanism of action based on the significant metabolite. Because when we do the database, we just check because when when you have the NMR, you need to have also the MS to support the identification. And also when you have those metabolites, the one you already identify, you need to go to see are they significant or not. And then when you for the mechanism, we suggest only the significant metabolites, and we can check their mechanism. When we compare between different groups and we see the metabolite, they are significant between, then we can put in the, mostly we are doing the metaboanalysis to see the mechanism, we put only the significant metabolite. And we can suggest the mechanism of action. And this is all suggestion, is not more, we cannot say it's very valid, but it's can have some clue to the mechanism. But if we,
which means further study need to be done. I mean, to verify these findings.
Well, we can check all the other metabolites by other studies. And as I say just now, if we do complementary for all the omics study is better, or one study is enough to draw a conclusion for that.
All right. Okay, we have another 2 questions. And we still have time, like 8 minutes for that. So the next question from Rohana, is there any potential study for us to study on the secondary metabolites that perhaps can inhibit COVID-19?
The secondary metabolite is very difficult to, especially the coronavirus study. It can be done on the animal or in the human, on the human. It's very difficult to check the secondary metabolite. Easy to, because we have the database and we have the library, we can see. But for the secondary metabolite, even if we give we feed the human, it's very difficult to trace the secondary metabolite because the concentration is very low.
So your suggestion maybe not specific secondary metabolite, so maybe in standard extract?
Metabolite, even if because the concentration is very low, it will synthesize other primary metabolites. The mechanism, it will be different. It's very difficult to trace that one is for that secondary metabolite. It's because of that secondary metabolite. We can just suggest based on the primary metabolite.
Okay. Thank you, Dr. Ahmed. So we move on the question from Ili Nadira. So her question is, can metabolomics approach distinguish between COVID-19 variety of strains, because we know different strains infected, at least in our population. So can we study metabolomics to differentiate between different strains and then compare to qPCR?
Yeah, this can be done by comparing different strain. We can just infect different animal by different strain, then we can see the differences and also see the biomarkers of each of the strain.
Interesting, because we know that different strains have different how fast or how slow it can infect, right?
It's very fast. It can be just like I say, the analysis and also sometimes the identification, because it's based on the database, also is very fast. But in the beginning, we need to build the model. If we already have the model about other strains, then just we need to have another model on the COVID-19, and then we can compare. It's very easy.
All right, thank you, Dr. Ahmed. So I think this will be our last question from Shen Hanli. So the question is, how feasible is it to translate findings from metabolomic study into the clinic? So do you think or do you envision that hospital laboratory will purchase NMR spectrometer in the future for COVID-19?
If we already build the model, and also in Malaysia, you already have so many research institutes. If have the model and also they already have the study, we can ask the hospital also to have their own NMR spectrometer, and also to the people who have the experience with that.
Okay, thank you very much, Dr. Ahmed. So I think this will be the end for our webinar series for today. But before that, I would like to thank to Dr. Ahmed Mediani for your willingness to, share this very insightful sharing and knowledge about metabolomics and COVID-19, and share how this metabolomics study can be done in terms of, the COVID-19 scenario, especially in Malaysia. And also, of course, I would like to thank to all the participants for your participation and also the question that you sent in the chat box. And before we end our sessions for today, I really hope that we can have a photography session. So could everyone please switch on or turn on the camera so that we can have a photography session. Okay, so basically I will say ready 1, 2, 3, and everyone can smile so that we can have very nice photograph, of course. So you ready, the moderator? Alright, okay. Our technical people ready, okay? So ready, 1, 2, 3. Okay, alright. All right, okay, thank you very much to all. So please wait until the end because we will send the QR code and also the link. Please scan the QR code or click the link so that you will get your e-certificate, and also for the UKM staff, your participation today will be recorded in ISPAL UK. So again, thank you very much for your participation, and hope to see you next year, inshallah. And Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Dr. Khalida, and all the participants.
Thank you, Dr. Ahmed.
Thanks.